Hi, everyone. So I want to start off a little bit with the science of this experiment. This is a challenge that we had. It's a relatively new experimental technique, and it presented some unique visualization challenges. And I'm going to go quickly through the science and then how we solved it. The idea behind the experiment is, how many of you are familiar with genetic interactions? Oh, there are slow hands going up. Okay. In a typical genetic interaction, you do single delete, double delete, and you measure the difference from expectation. In this case, you're doing a single delete. Let me get here. You're doing a single delete and then a point mutant. Okay? And so you're doing point mutants in a large structure. In this case, you're doing point mutants in histones, and then you're doing the gene knockout, and you're doing the cross there. So what you're trying to do is get genetic interactions that's actually probing at the structural level. Okay, so the challenge that we have with this is the typical visualization for genetic interactions is this nice, lovely clustered heat map. Okay, but the problem, if you look at this heat map, on the top are genes, and on the side are the residues, the point mutants. How do you find out anything from this that relates back to the structure? So that's the challenge that we were faced with. And it's also really difficult. The way that they were dealing with this previously is they would take this data, they had a Python script that would load the data in, that would process it, and then it would spit out a file that they would read into PyMol, which would generate a single image. And they would do this over and over and over again. All right, so our solution to this instead is to utilize a network representation. So in this network representation, we take all of the clustered genes, so they can browse those, a residue interaction network, edges, which represent the interactions, the genetic interactions between those genes and the residues, and then we integrate that with a 3D structural view, and the whole system is interactive, and they can take advantage of brushing and linking to go back and forth. This is what it looks like. So let me see if I can get my mouse over there. So the idea here is here are the genes. The genes are they're placed to represent the same as the heat map that they're used to. So this represents the clustering. Down in the bottom is the residue interaction network, and then as they click through, they can click a gene. That gene will follow the links to the appropriate residues, and then when those residues are selected, it will select and highlight the genetic interactions in the 3D structure. So we switch from a ribbon diagram to a space-filled model, and then we color that space-filled model by the genetic interactions. So with that, I will say there's the team. I won't go over it. This is a combination of Cytoscape and Chimera. So this is all desktop-based and all interactive. And come by and see me on my poster.
Thank you very much for this presentation.